Hi, everybody, and welcome to this week's lecture. What we're going to be looking at today is going to be genetics and inheritance. And we're going to be specifically looking at the passage of genetic traits from one generation to the next. So what we're going to try to accomplish is we're going to try to explain the basic patterns of inheritance. We're also going to be looking at a lot of vocab, specifically things like dominant and recessive, what those exactly mean. We're going to look at homozygous dominant, homozygous recessive, as well as heterozygous. We're also going to be able to infer an individual's phenotype based upon the genotype information that's going to be provided. We're also going to be able to predict outcomes from monohybrid crosses utilizing something known as a Punnett square. And then finally, we're going to be able to compare and contrast non-Mendelian genetics, such as incomplete and codominance, to Mendelian inheritance patterns. So let's go ahead and get started. The first thing we need to kind of talk about is genetics. What exactly is that? What does that mean? And why do we kind of care about that? Well, genetics are going to be really, really important. The reason why is because these are going to be our traits that we can physically see. These are going to be traits that are going to kind of define us and make us who we exactly are. These traits are going to be controlled by our genes, and those genes are going to be responsible for producing proteins, which are going to give us those traits. So some of the traits that are dependent on genetics are going to be things like attached earlobes or free hanging earlobes, eye color, hair color, skin color, things like that. So looking at our human genome, these are going to be the things that are going to dictate all of our genetics. Looking at this over here, this is going to be a picture known as a karyotype. So essentially what a karyotype is going to be is, again, a snapshot of somebody's genetics. Now, looking at this, you can see we have numbers 1 all the way through 22, and then we have these two at the very end. Now, 1 through 22 are going to be our autosomal chromosomes, meaning that these are going to be all regulated to our body. It's not until our last or our 23rd set, which is going to be our sex cells. So normal human cells are going to have about 46 chromosomes, typically 22 pairs of chromosomes plus 2 sex. So 22 autosomal plus 1, which is going to be our sex, giving us 23 pairs. Again, you can see each one of these has 2 of them in each slot. The only difference would be this one over here. This is XX, which is going to give us a female. If this was a male, it would look something like this. X with a piece here, and then Y with a smaller piece next to it. So these are going to be what we're talking about when we're talking about our genome, genetics, things like that. Now, when it comes to genetics, DNA structure and function, a quick recap is genomes are going to be the sum of an organism's DNA. So again, everything about that organism, everything that we need, our genetic information to build and maintain that organism. Now, there are going to be very specific regions known as genes, and these are going to contain instructions for building proteins. Now, these proteins are going to determine those traits, and these traits are going to be our distinguishing qualities or characteristics that are going to make you who you are. So looking at this over here, you can see this diagram. We have our chromosome right over here. And you can see that there's a ton of different bands. Those bands are going to be our genes. So you can see our examples, gene A and gene B. Now, again, these genes are going to be coding for our proteins. So you can see this region here is coding for protein A, and you can see this region over here is coding for protein B. You can see that those proteins are going to look drastically different. And the reason why is because those are going to be responsible for different traits. So a great example might be, let's say this could be coding for eye color, and this could give us brown, versus this might code for hair color, and this might give us black. Again, these are going to be our genes, which are going to code for proteins, which are going to give us our traits. And those traits are going to make us who we are. So now that we've kind of talked about exactly what our genes are and what everything that has to pertain to those genes, like our proteins and those traits, we need to take a little step back and see what people kind of thought of prior to discovering exactly what genes were. So before then, we didn't really know much about that. Here's a great example right here. In 1640, they kind of assumed that genetic material had little people in it, and that's kind of how these things were passed on. That's how people were made. We already had everything, and we kind of just were these little people that manifested and became, humans. It wasn't really until Gregor Mendel, which is known as the father of genetics, when he started to experiment and observe with these pea plants in order to kind of see what these genes are. And he made lots of unique and interesting discoveries when it came to those pea plants. He noticed that there were some characteristics that showed more often versus others. He was able to identify that if you took a certain plant and crossed it with another, you could end up with a purebred plant, which always produced the same kind of traits. And then when you cross that with another, plant with different traits, you end up with a unique percentage of these different types of traits. So let's go ahead and see exactly why Gregor Mendel studied these pea plants. One of the big things is they were very easy to observe. These pea plants over here had very unique and defining features that were very easy to indicate. Great examples would be things like flower color, seed shape, and seed color. Another reason is because you can get true breeding, meaning if you were to take, let's say, a purebred purple and cross it with another purebred purple, you would always get purple flowers. If you took a purebred white and crossed it with another purebred white, you would always end up with that same trait. Again, that made it very easy to kind of start off or give it a starting point. Male and female parts were in one flower, and they were very easy to manipulate during mating. And then each pea was a new individual. So again, he was able to evaluate thousands and thousands of offspring, giving him a massive sample size. Another reason is short generations. So again, it's much easier to do these experiments with organisms that grow and mature very quickly, because again, you're able to have a large sample size versus organisms that take very, very long. So in this case, Gregor Mendel was able to evaluate thousands and thousands of these versus if you were to use, let's say, mice or dogs or anything like that, which have a much longer lifespan and have a much longer maturing age. So he wouldn't have been able to have such a large sample size if he used something else. And then finally, they were very cheap and they were readily available. So again, giving him that ease of access in order to conduct these experiments. So let's go ahead and check out genetic crossing for one trait. So one of the things that we need to identify first is what a phenotype is. So phenotype, physical, is what you can physically see, or in this case, the physical appearance. One way I kind of remember this is P for pheno and P for physical. Phenotype starts with a P and physical starts with P as well. So phenotype is the physical appearance of the organism, so what you can physically see. So when it came to Gregor Mendel, again, one of the very first things he did was he identified true breeding. So in this case, whenever we have true breeding, you take an organism, in this case, the example I have is going to be a purple flower, and you cross it with a purple flower. Now, what you're trying to do is you're trying to have a purebred, so that means all of the offspring are always going to show or exhibit the same type of traits. So in this case, if you're taking a purebred purple flower, crossing it with a purebred purple flower, our F1, or in this case, our first generation, is going to give us all purple flowers. Again, if we cross it again, you can see in our F2 generation, or our second generation, all of these are also purple flowers. So in this case, you can see that we have a purebred flower color trait because we're starting with our P, parental generation, crossing those. We have our F1 generation, crossing that again. We have our F2 generation, and all of our organisms are purple flowers. Now, the reason why that was so important is because we wanted a purebred to start off these experiments with in order to see how these traits intertwine with each other and how these traits are going to be passed on from one generation to the next. So what Mendel did next was he went ahead and started crossing some of these parental plants to produce hybrids. So he was taking these pea plants, which had, in this case, a purple flower that was purebred, and he crossed it with a white flower, which was purebred as well. So in this case, this plant was pure, this one was pure as well. When you cross them, one thing he identified or noticed that in his F1 generation, this trait, the white flower, seemed to have disappeared. So it was no longer seen in the F1 generation. And then again, when you cross these, you can see in our F2 generation, we have 1, 2, 3 purple flowers, and then we have 1 white flower. So you can see that this trait has reemerged. So how can these observations be explained? So first thing is some variation of traits mask other versions of a trait. So this is going to be known as a dominant trait. So this is going to be a really good example because looking at this purple flower, you can see it's a purebred again. So the only genes that it has 4 colors should be purple. And we have a purebred white flower. Again, all the genes that this should have should all be coding for white flowers. When we cross these, you can see that the white flower gene has disappeared, meaning that our purple is much more dominant over our white. You can see that because, again, it's masking this, hiding this, and you can see that our phenotypes now are all purple flowers. So that would make our white flower over here a recessive trait. So our purple would be dominant, and then our white would be recessive. So although you can see that it's not going to show in our F1 generation, it is reemerging in our F2 generation. So in this case, that means we're taking these two purebreds and we're producing something known as a hybrid right here. And the reason why this is a hybrid is because we have a dominant trait in these right here, but they're also carrying that recessive trait as well. This is one of the reasons why you can see that we have that reemergence of that white. So this leads us to Mendel's first law of inheritance. So inheritance relies on the passing of genes from one generation to the next. So in the example prior, you can see that each true breeding parent carries 2 of the same genes for each trait. However, the version, or in this case, alleles of the genes may differ. For example, an individual can have 2 dominant traits, or 2 recessive traits, or 1 dominant and 1 recessive. So during gamete formation, when chromosomes number is reduced, each parent is passing only one version of this allele of each gene into each of their gametes. Okay, so this leads us to our terminology. So this and the next slide are going to be pretty important. So I would definitely start this and definitely look over what all of these mean, because there is going to be a lot of terminology when it comes to this. So I'll quickly kind of go over these. Monohybrid basically just means that we're crossing one gene. Okay, so we're crossing or only focusing on one type of trait, and that's where we're crossing, that's what we're going to track. True breeding means that we have parents that have the same trait. So again, if we go back to that purple flower, the first time Mendel kind of crossed everything, that was a true purebred, meaning that the only gene that that plant carries was the gene for that purple color. P1 is our parental generation. So again, that's going to be our very first, our very beginning. Our F1, which is going to be our first generation. So that's going to be the result of our P. And then our F2 is going to be the result of that F1 generation. And then self-fertilization is just crossing a plant with itself. A gene is just going to be a place on the DNA strand that's going to code for a specific trait. Remember, traits are characteristics. So again, things like flower color, eye color, hair color, skin color, things like that. Phenotype, remember, pheno starts with a P, is a physical trait of an organism. So something you can specifically see. A genotype is going to be the genes of that organism. So the genetic makeup, or in this case, the way I like to remember it is the letters that we're going to be using in order to identify all of the different types of genes. Homozygous means both alleles are going to be the same. So the example I have is PP or little p, little p. Homozygous, again, both are going to be the same. So Once we get into Punnett squares and we kind of start our crosses, you'll be able to see why we have these letters. But just remember, homozygous means both of our genotypes are going to be the same. Heterozygous means that the genes are going to be different. Okay, so again, when alleles from the same trait differ, so in this case, you'll have big P, little p. Our alleles, we have a dominant. Dominant means that it's going to mask all other genes. So that means if we have one dominant gene, that's the one that's going to show. The only way for a recessive trait to show is we need both lowercase letters, or in this case, both recessive traits. And then a zygote is the first diploid cell produced by fertilization. All right, so phenotype. Whenever it comes to phenotypes and genotypes, genotypes, remember, is the genetic makeup of an organism, and then an allele is going to be a different or alternate version of whatever we're looking at. So in this case, we're going to look at one pair of homologous chromosomes, and we're going to see what the genotypes could possibly be. So looking at this over here, genotype, this is going to be our homozygous dominant genotype. The reason why is because we have PP. Now, really quickly, when it comes to these letters, the way that we typically write these is, let's say we're looking at flower color, right? And purple is going to be our dominant gene. Now, whatever the dominant gene typically is, we take the first letter of that, and that's what we're using to identify that gene. So in this case, the color would be purple, and that's why we have a big P. Now, let's say the other one is white, and that's going to be recessive. Instead of using a W, we're going to use a little p to represent recessive. So in this case, for our color, dominant gene is going to be big P, and then our recessive is going to be our little p. So you can see this over here, our genotype. If we have two little letters, that's going to be homozygous recessive. Okay, so remember, dominant can dominate over all the other genes. So as long as there's one dominant gene, that means it's going to give us that trait. The only way we can have a recessive trait show is if we have two recessive traits. Okay, and then finally, this would be considered heterozygous. Hetero means different. So in this case, we have two different letters. We have a big B and a little b. So again, this is representing that we have two different types, heterozygous. All right, and that is going to conclude part 1 of lecture. As always, if you do have any questions, comments, or concerns, please feel free to reach out, and I look forward to seeing everybody in part 2.